Hi everyone, and welcome back to the Chapter 10 video lectures. In this lecture video, we're going to discuss the different types of DNA that forensic scientists use. So far in this chapter, we've been discussing DNA called nuclear DNA, or nDNA. This DNA comes from the nucleus of your cells. Forensic analysts can obtain DNA from multiple different types of cells, including skin cells and blood cells. Something that most people don't realize is that only certain cells in your blood contain a nucleus. As you may recall from the blood stain pattern lecture videos, red blood cells account for approximately 40% of your total blood volume. Red blood cells are like hollow frisbee discs, and they lack a nucleus in the middle. Forensic biologists instead use the white blood cells, which contain a single nucleus, to get nuclear DNA information. Now, white blood cells only account for approximately 1% of blood volume, which is one of the reasons that DNA extraction used to require a lot of blood to conduct. Small samples of blood found at the crime scene would only contain a few white blood cells and thus could not be used by investigators due to a lack of material to analyze. Nowadays, though, technological advances have allowed us to create a full or partial DNA fingerprint using really small samples. Instead of analyzing the material as is, scientists have learned how to replicate the DNA material that we find before they analyze it. This allows forensic biologists to create DNA fingerprints from much smaller samples than previously needed. One of the most common methods for DNA replication is called the polymerase chain reaction, or PCR. This technique is a relatively simple process by which the DNA is unzipped and replicated. First, the DNA is heated, which breaks down that double helix and it unzips the chains. Think about a ladder, taking a ladder apart by cutting down the middle of the rungs. The next phase is to rebuild each strand separately. New bases are added to the sample, and that rebuilds the ladder from each original piece. So you started with one ladder, cut it down the middle into two pieces, and then rebuilt a full ladder from each piece using new material. That leaves you with two ladders. Doing this over and over and over creates a chain reaction in which DNA is unwound, rebuilt, then unwound again, and process completed over and over and over until you can build exponential copies of DNA really quickly. What this type of duplication has allowed us to do is to analyze smaller and smaller samples while still getting a full DNA profile. In fact, DNA analysis has become so sensitive in the mid-2000s that we developed an analysis technique called touch DNA. Touch DNA involves the analysis of minute fragments of skin cells left behind when assailants touch victims, weapons, or anything else at the crime scene. Nowadays, scientists can get a full DNA profile from 7 or 8 skin cells by themselves. The reason for that is the replication process. By dividing and recombining the DNA strands over and over, even 7 or 8 cells can produce enough usable material for a full DNA profile. Even with touch DNA capabilities, though, we don't always get lucky enough to find cells with intact nuclei. This is where we use a less advanced technique that allows us to narrow down potential people, but not to individualize to a single person. Mitochondrial DNA, or mtDNA, is a type of DNA that's located outside of the cell nucleus. There are hundreds of copies of mitochondrial DNA in each cell, making it much easier to identify and collect than nuclear DNA. The problem is that mitochondrial DNA is only inherited from your mom, so you can't test mitochondrial DNA in the paternal line. You can only make comparisons to the maternal line. Another problem is that mitochondrial DNA does not usually change between parent and child. It doesn't mutate with the same regularity as nuclear DNA, so we can't use it to individualize to a single person. But we can use it to identify whether or not the mitochondrial DNA is consistent with the specific maternal line or not. Since it's not as useful as nuclear DNA, we use nuclear DNA when we have it. But in older and more degraded samples, we may not have any nuclear DNA to test, so that's when we turn to mitochondrial DNA. Mitochondrial DNA is also used for historical testing. For example, we've used mitochondrial DNA to identify soldier remains dating all the way back to the Civil War. When we test mummies or old skeletons, we often use mitochondrial DNA to confirm identification of the individual because nuclear DNA is not always recoverable from that old of a corpse. To give you an idea of how old and how degraded the bones can be, mitochondrial DNA has actually been able to be sequenced from the remains of a Homo sapien ancestor called the Homo heidelbergensis. These remains dated from almost 400,000 years ago. Scientists were able to recover the sample of mitochondrial DNA using only 2 grams of the femur bone. All right, well, that concludes the second part of Chapter 10. Join me in the final lecture video as we discuss some of the controversial issues relating to DNA collection and analysis.